All right, so starting with DNA replication, this is absolutely required when we are talking about cell division. So you need to replicate your DNA before the cell divides because we want to make sure that we have the same amount of DNA being passed on to all of the daughter cells. So DNA replication is absolutely necessary. And this begins with the unwinding of the DNA of the double helix structure at the origin of replication. And DNA replication occurs during this DNA synthesis phase and interphase of the cell cycle. Elongation creates a replication fork in the DNA. And so if you're looking here, if DNA is unzipping this way, this is done through helicase, you'll see that there will be replication via DNA polymerase from the 5' to the 3' end. So remember I was telling you that there is this directionality with a nucleic acid sequence. The enzyme DNA polymerase cannot add on nucleotides from the 3' to the 5' end, only from 5' to 3', and this is based on the chemical structure of the nucleotides. And so this leading strand, you can see if the helicase is unwinding this way, the DNA polymerase can basically just follow it along. However, as the helicase unwinds this, DNA polymerase basically needs to add on from 5' to 3' ends these Okazaki fragments, and those need to be ligated later on in the DNA replication cycle. So this is called the lagging strand because it takes a little bit longer. And then finally we have termination. So because DNA is linear and because of the way that the DNA polymerase works, you will always lose a little bit of information at the end of every replication cycle. And telomeres are bits of repetitive DNA sequence at the end of our chromosomes that prevent the loss of important genetic information. And this is really important in cancer biology and aging biology, but we won't get more into it here.